Let's revise KCSE 2023 Biology Paper 2. On to our first question. The following diagram represents a section of the mammalian respiratory system. Part A. Identify the region of the mammalian skeleton where the represented section is found. So in short, they're simply asking us, where is this part of the body located? And this is found in the thoracic region. Roman 2, the part labeled F. So the part labeled F, this is the pleural membrane. Part B, explain the function of the part labeled F. So the function of the pleural membrane is that it secretes the pleural fluid. This reduces the friction of the lungs during breathing. Moving on to part C, explain the structural adaptations of the parts labeled E and G to their functions. Okay, let's look at the diagram. E refers to the trachea and G is pointing at an alveolus. Now, if we start with the trachea, number one is that it has C-shaped rings of cartilage to prevent it from collapsing. Another adaptation is that it also has cilia. So the cilia waft in bits in order to move mucus and any foreign particles within it towards the pharynx for removal. It also has goblet cells which secrete mucus. Moving on to the alveolus, number one, they are moist. Why? So that the respiratory gases, that is oxygen and carbon dioxide, can dissolve and diffuse in solution form. Number two, they are highly vascularized to maintain a steep concentration gradient for faster rate of diffusion. Last one, they have a thin epithelial membrane, which is only one cell thick. And this is so as to reduce the distance across which the diffusion of the respiratory gases occurs. Part D. Give similarities between the part labeled G and gill filaments. Okay, let's just consider that. What do the alveoli and gill filaments have in common with one another? They are both respiratory surfaces. One is found in mammals and the other in fish. So they are going to have adaptations that are similar to one another. So both are moist, both are highly vascularized, and they have a one cell thick epithelium. And another one actually is that they are both numerous. So these are the similarities that they have in common with each other and with other respiratory surfaces. Moving on to question 2. In an investigation, data on students' heights and blood groups was collected and presented in graphs H and J as shown. So we are having two graphs, one based on the height of students and the other on blood groups. So A Roman 1, state the type of variation illustrated by the graph H. Now, when we talk about variations, these are simply differences that are present in members of the same species. For example, the differences that are present among humans are what are termed as variations. Now, variations are of two types. We have continuous variation and discontinuous variation. Now, when we talk about continuous variation, an example of which is height in human, you're going to have a continuous gradation between two extremes. So you're going to have a case whereby few are extremely short, again a few more are extremely tall, but majority of humans are going to fall between the intermediate height, near there. So you're going to have continuous variation. You cannot just simply take people and group them, say that according to your height you belong to this category or such. It doesn't work like that when it comes to continuous variations. Another example is that of pigmentation of the skin. Now, when it comes to discontinuous variation, you belong in one group or another. That's it. So a good example is the ABO blood group system. You're going to have an individual belonging into one blood group only. So it's AB, ABOO, nothing else. So if we look at this graph H, this is that of the height of students. Of course, this is going to be continuous variation. Roman 2, give a reason for your answer in 2A. Now, the reason is because you have a wide range of variations. Now, this type of curve is known as a normal distribution curve. Part B, explain the advantage of having the greatest proportion of students with blood group O as illustrated in graph J. Now, actually, the majority of the human population are members of blood group O. And the reason for this is because blood group O individuals are universal donors. So in case whereby there's a need or an emergency, they can donate enough blood. Moving on to part C. After the investigation, a student of blood group O reported that the father and mother are blood group A and B respectively. Use a genetic cross to illustrate this possibility. Okay, so we have two parents, the father belonging to blood group A and the mom B. Now, curiously, their child has blood group O. So we are supposed to, one, identify the genotypes of the parents so as to illustrate how this could even be possible. Now, when it comes to genotypes, we are going to have two alleles present. We already know one of the alleles present in the father and mother's genotype. So in the case of the father, he definitely has the A allele. In the case of the mom, the B. So what is the second allele going to be? O, of course. Why? This is because when it comes to blood group O, the O allele is recessive and can only be expressed if it's in homozygous state. So what I'm simply meaning is that when it comes to the student having blood group O, that means the genotype of this student is OO. So the student would have gotten one of these alleles from the mom and the other from the dad. Therefore, the parental genotypes are AO and BO. So moving on to the crossing, and there you have it. Question 3. In an experiment, students soaked maize seeds in water for 48 hours at room temperature and tested them for reducing sugars. Part A. Name the reagent the students used to test for the reducing sugars. This, of course, is Benedict's solution. Part B. Explain the reasons for soaking the seeds. Soaking the seeds softens the seed coat, and this is important because it allows entry of water. Water is needed to activate enzymes. These are the enzymes that are going to hydrolyze the starch. They're going to break down the starch to reducing sugars. Part C. Set the observation made by the students during the food test. The blue color changes to yellow, orange, or brown. Now, I want to say this. You can simply phrase your answer as blue color changes to yellow or blue color changes to orange. If you decide to include the sequence, then ensure that it's correct, because otherwise you will lose marks. Part D. Explain the effect of soaking the seeds in hot water on the food test results. Now, hot water is at a high temperature. So what will happen is that when it comes into contact with the seed, it's going to denature the enzymes. So if the enzymes are denatured, you're not going to have hydrolysis of starch. Starch is not going to be broken down to reducing sugars. And therefore, you're going to get negative results. What do I mean by negative results? You're not going to have any color change. The color remains blue. E. A sample of maize seeds were planted in soils with favorable conditions but failed to germinate. Suggest possible causes of this failure. The seeds here are clearly dormant. Now, when it comes to dormancy, it can result from a variety of factors, some of which are from the environment, environmental conditions, and some are internal factors, present within the seed. But in this case, we have been specifically told that the conditions were favorable. So that means these are the conditions that are present internally within the seed that are affecting its germination. So number one, the seed could be inviable, or the embryo could be immature. Or maybe you have low hormonal concentration. The hormones that are responsible for promoting germination are low. Or maybe you have presence of germination inhibitors. Question 4. In an investigation, students placed dry cobalt-2 chloride paper on both sides of a mesophyte leaf and covered the strips with sellotape as shown in the following experimental setup. Okay, so we have cobalt-2 chloride paper placed on a leaf. Okay, guys. Cobalt-2 chloride paper. What happens is that when it's exposed to moisture, it turns from blue to pink. So we already have a hint of what to expect. We are testing for moisture, for the presence of water. And since it has been placed on the leaf where stomata are, that means the aim of this experiment is to investigate the rate of stomatal transpiration. You have to include the term rate because we want to find out, is stomatal transpiration occurring faster on the upper surface of the leaf as opposed to the lower surface? So we are not just interested in finding out transpiration is taking place, but we want to know, is it different in these two surfaces? B. Roman 1. State the observation the students made on the cobalt 2 chloride paper. So what will happen is that transpiration will take place, releasing water vapor. This of course is going to turn the cobalt 2 chloride paper from blue to pink. Roman 2. Account for the difference in the time taken for the observations to be made on the two cobalt 2 chloride papers. Okay, now let's first agree. Where is transpiration going to occur faster? Is it going to be on the upper surface of the leaf or the lower surface of the leaf? Yes, it's going to be on the lower surface of the leaf. So in the lower surface of the leaf, we are going to have a faster rate of transpiration. That means more water vapor is going to be lost. Therefore, you're going to have a faster change of color in the cobalt 2 chloride paper that is attached to the lower surface. Why? Because this is where you have a higher number of stomata. More stomata are located on the lower surface of the leaf compared to the upper surface of the leaf. Part C suggests two modifications that students would make on the setup to have the observations made within a shorter time. So they're just simply asking us, how can we speed up the rate of water vapor that is released to the environment? So you can increase the amount of water in the pot. You can increase the air currents, maybe by introducing a fan or by actually fanning the plant. Remember, wind increases the rate of transpiration. Or you can even increase the temperature. Temperature, wind, and light intensity increase the rate of transpiration. So the higher these are, the faster the rate of transpiration and the faster the change in color. By the way, if transpiration is a topic that is a bit challenging for you, please go check out my videos. I have 3 videos explaining in detail what transpiration is and the environmental and structural factors that affect the rate of transpiration. Moving on to question number 5. The following diagram represents the structure of a human eye. Part A. Name the part labeled M. This is the pupil. Part B. Explain the adaptations of the part labeled L to its function. Now, if we look at L, this is the cornea. So it's transparent to allow the passage of light. It's curved outwards to allow refraction of light. You know, this is bending of light. Remember, light is coming from the atmosphere and passing into the eye. So whenever light moves from one medium to another, it's going to be refracted. Part D. State the role of tears in the human eye. So the tears moisten the eye. They wash off any dust or foreign particles that are present in the eye, and they are also antimicrobial. So this means that they contain an enzyme called lysozyme that kills bacteria. Proceeding to question 6, during an ecological study, students obtain the following data from a certain ecosystem. Okay, so we have the trophic level of organisms and the number of organisms found in each trophic level. So for example, the producers are 1,000, primary consumers 200, secondary 30, and the tertiary consumers are 4. So you are expected to use the data in the table to draw a pyramid of numbers on the grid provided. And there we have our pyramid of numbers. Now, as you can see from the question, this is worth 5 marks. So how are the marks distributed? Number 1 is for having the correct pyramid, you know, for having an upright pyramid. Another mark is for the scale, having a correct scale. Third mark, proportionality, having a width that is consistent throughout. Fourth mark is for plotting. You should ensure that all the values have been entered correctly. And last one, for labeling. Part B, account for the shape of the pyramid. So this is an upright pyramid, you know, whereby the base is wide. So you're going to have the highest number of organisms being producers with the lower numbers being the consumers. So this is important because it allows support of the consumers, you know, in terms of nutrients. So the numbers of organisms decrease progressively as you move from one trophic level to another. And the reason for this is because you're going to have energy being lost as you move from one trophic level to another in different forms such as excretion, respiration, and so on. Part C. If the following organisms were found in the ecosystem represented by the data, draw a food chain to illustrate the feeding relationship in this ecosystem. So we are going to start with the producers, which in this case are the plants. The plants are being fed on by the mouse, and then the snake, and lastly the eagle. Moving on to Roman 2. Explain the effect of a severe drought on the population of organisms in the ecosystem. So what will happen is that the producers, which in this case are the plants, are going to dry up because of the lack of water. So this will reduce the food that is present. So it's going to cause starvation of the mice. Why? Because their food source has been reduced. And of course, the organisms that follow afterwards are also going to be affected. So you're going to have the snakes and the eagles also affected. So the organisms are either going to die or they're going to migrate, move to other favorable habitats. This, of course, will ultimately cause a decrease in the population of the organisms in the ecosystem. Roman 4. How would predation by snakes lead to the emergence of new species of mice in the ecosystem? Okay, let's start with the mice. By the way, this question is simply about natural selection. So mice have variations. These are the differences that are present within this species. So these variations are going to be either advantageous or disadvantageous. Advantageous variations are those that are going to make these mice, some of these mice, better adapted to survive in their environment. Unfortunately, those that have disadvantageous characteristics are going to be poorly adapted and therefore are going to be preyed upon. Those that have advantageous variations are going to survive and reproduce and give rise to offspring with similar characteristics. Over time, this accumulation of variations is going to lead towards the formation of a new species. Part D. Describe the method one would use to estimate the population of a particular plant species in the ecosystem. Now you can use a quadrat. Number one, you're going to measure the area of the study in meters square and then throw the quadrat randomly in the area of study. So all the individual plants that are present within this quadrat are then going to be counted and recorded. You're going to repeat these several times, 3 or more times, and the totals in each throw is going to be summed up, then averaged. Multiply the average with the total area of the study to get the approximate total population of the plant in the habitat. Our last part, the essay question. So question 7 is divided into two parts. Part A, describe the various functions of lipids in the human body. Part B, describe how the process of photosynthesis occurs in green plants. Question 8. Describe how the section of the human digestive system from the mouth to the stomach is adapted to its function. And those are the answers.